Hi friends, hope you are fine. We have already discussed 15 differences between plant cell and animal cell. In this video, let us focus on the difference between cytokinesis in plant cell and animal cell within 5 to 10 minutes. Cytokinesis, the word meaning is cyto means cell, whereas kinesis means movement. Actually, it is a cytoplasmic division after mitosis or meiosis. As you see, this is the cell cycle. After cell cycle, after the mitotic phase, two daughter cells are formed, and these cells should be separated, and both the cytoplasm and the genome should be equally distributed to these daughter cells. And that process is called the cytokinesis. It can be defined as the cytoplasmic division of a cell at the end of mitosis or meiosis, bringing about the separation into two daughter cells. Now let us understand the cytokinesis in animal cells. In animal cells, the first step is the positioning and formation of a contractile ring. As you see, this is the contractile ring, this red dotted line. It is formed of actin and myosin filaments. And the positioning of this contractile ring is guided by these central spindle microtubules. So these microtubules are involved in positioning this actomyosin contractile ring at the center of the cell. Step 2 is cleavage furrow ingression by actomyosin contractile ring. So as the result of movement of actin and myosin filaments, a cleavage furrow is formed, an ingrowth that causes the constriction or ingrowth forming a deep cleavage furrow. Then at a later stage, at the end of cytokinesis, a special structure is formed. An intercellular bridge is formed between the two separating cells, daughter cells, with a specialized transient structure, a temporary structure in animal cell, which is called as the midbody. This midbody is made up of tightly bundled antiparallel microtubules. So it's a transient structure that connects these two daughter cells at the end of cytokinesis in animal cell. It mediates the final step, which is called the abscission. It also is involved in localizing the site of abscission. And the final step is abscission, forming two daughter cells. So this midbody is responsible for localizing the position of site of abscission. Then there are many protein complexes like ESCRT3, which is expansion is endosomal sorting complex required for transport, which is a protein complex that is required for abscission, that helps in abscission. So this filament surrounds this midbody and helps in abscission process. So cell division ends with the cutting of microtubule and this intercellular bridge connecting the two daughter cells at specific locations, or that is determined by this midbody. Finally, the separation of these two daughter cells is called as abscission. So these are the steps involved in the cytokinesis of animal cell. Now let us understand the cytokinesis in plant cells. Cytokinesis in plant cells is completely different, as we know that plant cell is with cell wall, so the process is entirely different. Step 1 is the formation of phragmoplast, which is a transient temporary structure that is formed in plants at the end of a cytokinesis. This structure is made up of microtubules and actin filaments, and that forms or acts as a framework for the formation of cell wall or cell plate, finally separating the two daughter cells. So this is the structure, phragmoplast. As you see, it is made up of actin filaments and also microtubules. Now the second step is transport of Golgi derived vesicles with cell wall materials. So this cell wall materials pinches off from Golgi apparatus as loaded in vesicles, and that is transported to this central region. So this is actually gated by this phragmoplast, microtubules on the phragmoplast. So it aligns as a center. These are cell wall materials that are loaded in vesicles that aligns at the center. So this is the electron micrograph, as you see. So this is the cell plate in the middle. Then this is a phragmoplast. So this phragmoplast actually serves as a scaffold for placing this cell wall material in the precise location, in the correct location. So this is the separating nucleus of the first cell, and this is the nucleus of the second cell, daughter cells. In the next step, vesicles fuse to form a cell plate. Then this vesicles fuses and this grows and finally reaches this plasma membrane. Meanwhile, plasma membrane is formed on both daughter cells internally, and this is the cell plate. Cell plate or cell wall finally forms on. Meanwhile, plasma membrane of both daughter cells are formed. These vesicles or membranes of these vesicles also take part in the formation of plasma membrane and finally separating these two daughter cells. So this is the cytokinesis in plant cell. Let me summarize the cytokinesis in plant cell. Step 1 is the formation of phragmoplast. That is a structure that acts as a scaffold for the deposition of cell wall materials that is provided by the Golgi apparatus as vesicles that reaches the center, and finally it fuses to form a cell plate. During that time, plasma membrane is formed on both daughter cells and finally forming a complete cell plate or cell wall separating the two daughter cells. Hope you understand the cytokinesis in plant cell and animal cell. Take care, stay blessed. Thank you so much for your support. You are with biologyexamsforyou.com.